Friends, welcome to Distinction Learning Platform, and in this video we are looking at Biology 2017 Paper 2. And before we begin answering this question, make sure that you comment in the comment section as well as you like the video. And in the comment section, you'll find a link that will take you to the website where you'll find now some other subjects like biology, chemistry, physics, and many other subjects. Alright, make sure that you click on that. Let us begin. So this question came under section A, question 2, and the question reads, Figure 2.0 shows a graph showing an enzyme catalyzed reaction. So this is the graph that we have been given, all right? And the first question is talking about this one. What is an enzyme? And it carried about 1 mark. So what is an enzyme? So remember that an enzyme, this is just a biological catalyst. So I'm going to say an enzyme is the biological catalyst. Okay, let us use this catalyst that speed up the rate of chemical reactions without itself being chained. This is what we call an enzyme. So let us look at this definition. We are saying it's a biological catalyst. Why are we saying it's a biological catalyst? Because an enzyme, it is a substance that can be found inside a living organism. That's what we call an enzyme. Alright? Because there are also some catalysts that can be found outside the living organisms. Okay, and we call them, those are chemical catalysts. But biological catalysts, these are what we call enzymes. Why? Because they can be found within living organisms or inside living organisms. So this is the basic definition of an enzyme. And an important notice that you need to take note is that these enzymes, they always speed up the rate of chemical reactions. They make sure that this chemical reaction that happens in our body, they are going to be faster, or they are going to finish faster. Okay, that's the role of an enzyme. Okay, and the other important point that you need to take note is that apart from speeding up the chemical reaction, the rate of chemical reaction, these enzymes, they are not going to be chained. Okay, what does this last statement mean? It means that if A is an enzyme and C is something else, okay, C is a substrate. So this enzyme, before a chemical reaction and after a chemical reaction, is not going to be changed. It will just remain the same. But this substrate, it is going to change maybe from C to D. That's what it means, not being changed throughout the chemical reaction. So that is question A. So now let us go to question B. Question B is saying, what is the optimum temperature of the enzyme reaction shown in the graph in figure 2.0? Okay, so again the question reads, what is the optimum temperature of the enzyme reaction shown in the graph in figure 2.0? Okay, now the key point here that you need to understand is what? Optimum temperature. Optimum temperature, this one, this point. Okay, optimum temperature. What is optimum temperature? Someone who doesn't know that the optimum temperature is that, or the definition of optimum temperature, basically they are going to fail answering this question. Now let us understand about what we call optimum temperature. What is optimum temperature? So optimum temperature, according to enzymes, is the temperature where enzymes work the best. So what is optimum temperature? We're saying optimum temperature is a type of temperature where enzymes are going to work the best. And remember that optimum temperature in the body is going to be around 37 degrees Celsius. That's where enzymes work the best. So here, from the graph, because the question is saying we need to use the graph in order to answer this question, so we're going to use this graph so that we can understand or obtain that optimum temperature where enzymes work the best. So if you look at this graph, from this side, it is from the y-axis here, it is saying this is the rate of reaction. On the x-axis, this is the temperature. So we're going to pick a single number on the temperature side here or on the x-axis, and we're going to call that as optimum temperature. Now the question that you can ask is, how do you find the optimum temperature? All right, so you come on the graph here, and where the graph is turning, the maximum point of the graph, or where the graph is turning, that's where you are going to find the optimum temperature. Now let us follow. So this is the graph. So the graph is turning around this point. This is where the graph is turning. So then you mark a point there. Then you drop and up until you reach the x-axis. Okay, and that will be your value for optimum temperature. Again, we go back. How do you find the optimum temperature? You go on the graph, and where the graph is turning, you mark the point there, then you start dropping like that. And the moment you reach the x-axis there, that will be your value for the optimum temperature. Okay, so if this is 35, as you can see, this is 35. Basically, after 35, you're going to have 36 and you're going to have 37. Okay, so now remember what I said. What is the normal optimum temperature in our body? That is 37. So this is 35, then followed by 36, then there will be 37 there. I need to count, but basically it has to be 37. Okay, so our optimum temperature here, it is what? 37. And don't just write 37, make sure that you put the units for temperature, which are degrees Celsius. So we're going to say 37 degrees Celsius. All right, that's what we call optimum temperature. We proceed. Then question B, Roman numeral 1. Okay, it is saying, at what temperature in figure 2.0 is the enzyme completely denatured? So biology questions, you need to be understanding them. If you don't understand those questions, basically also the way you're going to answer is going to be wrong. So what we're saying is that, what are the keywords that you can obtain from this question B1 or question B Roman numeral 1? So there are some what can I say, some keywords. So let us highlight those keywords, like this one, completely denatured. Because if you don't understand completely denatured, basically you get this question wrong. And they need the number here, because they are saying at what temperature. Basically they need a figure. Okay, so now at what temperature in figure 2.0 is the enzyme completely denatured? Let us understand completely denatured. What does it mean? Denaturing under biology, we mean is like dying. Okay, if I say an enzyme has been denatured, I mean to say the enzyme is dead. Okay, that's what it means. So denaturing basically it means dying. Okay, so now from the question, from this statement, or understanding this statement, basically they are saying, where are we going to see that enzymes now are completely dead? Where? So from the graph, as you can see, starting from 0 up to the optimum temperature, this is how the enzymes are working. But after 37 now, as you can see, the graph is like decreasing like that. So this decline of the graph, it means that the enzymes are being denatured or the enzymes are dying. So now, completely, they are saying, go to the graph where now the curve is at 0. So after optimum temperature here, you come here, okay, where the graph now has dropped completely, where it's somewhere around here, which is 60. So 55 you have this. So if you look at 55 here, if you look at 55 is somewhere here, where this graph at least it has some value for y-axis. But where this curve or this graph is completely ending, is ending at what? 60. So that's where the enzymes are now completely denatured. So it's at what point? 60 degrees Celsius. 60 degrees Celsius. So that's how you can find the temperature where enzymes are completely denatured. Just go on the graph and go where the graph is ending. That is it. The enzymes be denatured completely. All right. So now let's go to Roman numeral 2, question B. Okay. Then they are asking you, give a reason for your answer. So again, we need to understand the question. Give a reason for your answer. They are telling us, or they are asking us to say, why do we say that the enzymes are dead at 60 degrees Celsius? Why do you say like that? So now we need to go back to how temperature affects enzyme activity. So if explain how enzymes denatures, or how enzyme make sure that how temperature makes sure that the enzymes are what? Are completely denatured. So we go back to the concept that we learned. Okay. So let us try to understand this point. If you look at an enzyme, the structure of an enzyme, this is an enzyme. So I'm just picking N. So let us pick this one. All right. If this is an enzyme, all right, and remember that an enzyme is going to have a lot of parts. So let us look at the parts of an enzyme. So we've got this part. So let us just write that one. So we've got this part. Okay, this part is what we call an active site, and this part outside is what we call the allosteric site. Allosteric site. Okay, so the enzyme is going to have two important sites. There will be an active site and what? The allosteric site. So this site where the substance is going to fit, or where nutrients and other things are going to fit into an enzyme, we call it an active site. And where the substrate are not fitting into an enzyme, we call it an allosteric site. Now, what happens is like this. So if you are increasing temperature more than the normal body temperature, which is more than 37, basically the temperature that you are applying, this temperature, high temperature. So let's just say high temperature, high T. This temperature is going to start making sure that this active site is going to lose its shape. So in short, what we are saying is that the temperature, how does it affect the enzymes? Or to be completely denatured, they are going to change the shape of an enzyme. If this shape is going to be changed because of high temperature, the active site is going to be lost, as well as the allosteric site is going to be lost. In that, showing that all those nutrients that's supposed to fit in the active site, they are not going to be fitting there. So the correct way, or the correct reason for this one, is that high temperature causes change in the what? In the shape of an enzyme, including the active site as well as the allosteric site. Okay, so I'm going to say the reason for this is that because high temperature, okay, what did we say? Changes the shape of an enzyme. So let's just make it simple. We say changes the shape of an enzyme. The shape of an enzyme. Okay, so this is the basic reason why the high body temperature, more than 37, is going to make sure that the enzymes are what? Completely denatured. Why? Because this shape is going to be changed because of high temperature. That's how temperature makes sure that there is completely denature of what? Enzymes. So that is question B2. Let's go to C. Explain why it is important to measure the body temperature of a sick person. Very simple question. Now, why do we measure the body temperature? Okay, so the body temperature, the reason why we measure the body temperature is to make sure that to detect fever. Okay, so to detect fever. So why do we measure the body temperature? So I will explain what it means when you say fever. Okay. So why do we measure the body temperature? So the first reason is to make sure that we detect what? Fever. Okay, so let's just say in simpler terms, let us just say to know if there is high body temperature or low body temperature. So in brackets, I'll say fever. Detect fever. Either way. Now, let us understand what it means, fever. What is fever? Fever basically, it is an observation or something, it is a symptom that's going to be felt by a person where they are going to have what? High body temperature. Okay? The moment someone is going to have high body temperature, basically more than 37 degrees Celsius, basically those patients or people, they are going to have fever. Okay, what is fever? Fever basically is high body temperature more than the normal body temperature. That's what we call fever. The reason why we measure the body temperature is to make sure that you know if someone is having fever or not. That's the main reason why we detect or we measure the body temperature. That's just the normal reason. Or you can just say to know if there is high body temperature or low body temperature. That's all. You can say something like that. Alright? Let's go to question C2. Explain the ways in which the body temperature of a sick person can be brought down to normal. So they're just asking us to say, if someone is sick, what do you do? Okay, because if someone is sick, it means that the body temperature now is not at 37 degrees Celsius. It's either 38 or maybe 35 or 36. Okay. So now, what do we do if someone is sick? The first important thing that we do is to give medication. Okay. So the first thing that we do is to make sure that give medication. You give medicine. So you give what? Medication. Okay. So maybe if someone is having like high fever, basically you are going to give like paracetamol, you give anti-inflammatory drugs, a lot of drugs that are going to be given if someone is having high fever. But the medication, they will depend on what the disease the people they are having. Okay. Then we go to question reason number 2 or way number 2. Okay. Apart from giving medication, what do you do to a sick person? Basically, we give supportive care. Whatever they need, we need to be there. You give like water, make sure that they bath. Okay, a lot of ways. So those things we can just simplify them in what? Giving supportive care. So the other important thing for you to make sure that the body temperature of a sick person is brought down to normal, we need to make sure that we give supportive what? Give supportive care. Supportive care. Basically, we give water whenever they need water if they are dehydrated. So let's say hydration. So let's just put here in hydration. Hydration means that you give them water whenever they are thirsty. That's what we call supportive care. Okay? All the best. Make sure that you study more. Until next time.